For today's video, we will talk about the chapter 17 of the book of Histology. Chapter 17 is entitled Hepatobiliary System and Pancreas. But first, let us define what is hepatobiliary system and what is pancreas. Hepatobiliary system, or also referred to as the biliary tract or system, is very essential for digestion. It is made up of the liver, which produces and secretes bile. And then we have the intrahepatic bile ducts, or IHBDS, which are a network of small tubes that carry bile inside the liver. And then we have the extrahepatic bile ducts, EHBDS, which are small tubes that carry bile from the liver and gallbladder to the small intestine. And lastly, we have the gallbladder, which stores the bile from the liver. So, the organs that I have mentioned, which are the liver, bile duct, and gallbladder, makes up this hepatobiliary system. Now let's proceed to pancreas. What is pancreas? Pancreas is an organ and a gland. Note, it is an organ and a gland. Glands are organ that produces and releases substances in the body. The pancreas performs two main functions. So pancreas has two main functions. These two main functions of pancreas is the exocrine and endocrine functions. For the exocrine function, pancreas produces substances which is the enzyme that helps with digestion. For its endocrine function, it sends out hormones that control the amount of sugar in our bloodstream. These hormones are the insulin and glucagon. So, as said in this note, pancreas has two main functions, the exocrine and the endocrine. For its exocrine, it produces substances which is enzyme, and for its endocrine, it sends out hormones which is the insulin and glucagon, which control the amount of sugar in our bloodstream. So, to better understand hepatobiliary system and pancreas, come on, let's dig deeper into it. Okay, let's begin with the liver. The liver is the largest gland of the body, situated mainly in the right hypochondrium below the right dome of the diaphragm in the abdomen. The liver may be regarded as a modified exocrine gland that also has other functions. The liver substance is divisible into a large number of large lobes, each of which consists of numerous lobules. As you can see in the first picture, that is the illustration of the liver. And beside it is a schematic representation of a classic liver lobule. This classic liver lobule is made up of central vein, portal triad, liver cells, and sinusoids. The round shape in the middle of the liver lobule is what we call a central vein, while the colorful shape inside the triangle is what we call a portal triad. And this portal triad is made up of portal vein, which is the blue rod shape, hepatic ductule, which is the green circle one, and the hepatic artery, which is the red one. Lastly, the liver lobule has a sinusoid, which are the spaces located between the liver cells. So, as you can see, that is what the schematic representation of a classic liver lobule looks like. Now, let's proceed to its microscopic features. The liver is covered by a capsule made up of a connective tissue. And this connective tissue that covers the liver is what we call a Gleason capsule. As you can see below the illustration of the liver, there is an arrow pointing to the surface of the liver. That is what we call a Gleason capsule. Gleason's capsule covers liver surface as an extension of peritoneum except the bare area attached to the diaphragm. Glycan sheath is defined as glycan capsule surrounding the intrahepatic portion of the hepatic portal system. Glycan's capsule or sheath continues into the subperitoneal space of the gastrohepatic and hepatoduodenal ligament in the one direction. It also continues into the subperitoneal space of the ligamentum teres and falciform ligament in the other direction, curved arrows. Now, let's proceed to hepatic lobules. In sections through the liver, the substance of the organ appears to be made up of hexagonal areas that constitute the hepatic lobules. In transverse section, each lobules appear to be made up of cords of liver cells that are separated by the sinusoid. As I've said earlier, the sinusoids are the spaces between the liver cells. However, the cells are really arranged in the form of plates, one cell thick, that branch and anastomose with one another to form a network. Spaces within the network are occupied by the sinusoid. That is the spaces between the liver cells. Now let's go to the portal canal. Along the periphery of each lobule, there are angular intervals filled by connective tissue. These intervals are called portal canals. The canals forming a connective tissue network permeating the entire liver substance. So this canal contains a branch of the portal vein, a branch of the hepatic artery, and an interlobular bile duct. These three structures collectively form a portal triad. So, as I've said earlier, liver lobule has a portal triad. And it is made up of portal vein, hepatic ductule, and hepatic artery. So under the picture of the liver lobule, you can see a zoom-in photo of the portal triad. And as I've said earlier, the rod shape is what portal vein is. That is its structure. And then the green one is the hepatic ductule, and then the red one is the hepatic arteriole. So blood from the branch of the portal vein and from the branch of the hepatic artery enters the sinusoid at the periphery of the lobule and passes towards the center. Here, the sinusoid opens into a central vein that occupies the center of the lobule. The central vein drains into hepatic veins. Blood vessels and hepatic ducts present in portal canals are surrounded by a narrow interval called the space of Moll. Now let's proceed to portal lobule. But first, observe the picture you see on the left side. The picture that you have observed shows the concept of portal lobules. Portal lobules are the pink inverted triangles shown in the illustration, and the shaded green ones are the hepatic lobules. In this case, each functional unit is a triangle having a central axis through a portal field and imaginary vertices through the three different but closest portal canals surrounding it. The area covered by the triangle represent the hepatic regions that secrete bile into the same bile duct. And also note this, that portal lobule is made up of parts of three hepatic lobules. These three hepatic lobules are the centrilobular, surrounding central vein, mid-zonal, and lastly, the periportal adjacent to portal tracts. That is portal lobule. Now let's proceed to the next. But first, observe the picture you see on the right side. The picture that you have observed on the right side shows the concept of the portal acini. Portal acini is the pink diamond you see in the illustration. Portal acinus is a still smaller unit which consists of a diamond-shaped area of liver tissue supplied by one hepatic arteriole. The red circle you see in the illustration is what we call a hepatic arteriole, running along the line of junction of two hepatic lobules. So the hepatic lobules, as I have said earlier, are the shaded green ones. Then two central veins lie at the end of the acinus. As I've said, the diamond one is the acinus, and in each end, it has two central veins. The circle one is the central veins. In this part, we will gonna learn about the liver in a panoramic view. As you can see on the left illustration, it shows the liver as seen in drawing, while on the right side, it shows the liver in photo micrograph. The panoramic view of liver shows many hexagonal areas called hepatic lobules. As you can see in the illustration, there are so many hexagonal areas, and these hexagonal areas are what we call hepatic lobules. And these lobules are partially separated by connective tissue. Each lobule has a small round space in the center. This is what we call the central vein. So, look at the picture. You can see there is a round space in the middle of the hepatic lobules. These round space are what we call central vein. A number of broad irregular cords of cells seems to pass from this vein to the periphery of the lobule. These cords are made up of polygonal liver cells. These polygonal liver cells are what we call hepatocytes. Along the periphery of the lobules, there are angular intervals filled by connective tissue. Each such area contains a branch of the portal vein, a branch of the hepatic artery, and an interlobular bile duct. So these three that I have mentioned, the portal vein, the hepatic artery, and the interlobular bile duct, constitute a portal triad. And let me tell you this, we can recognize a liver tissue if we identify hepatic lobules and portal triads because it is enough for us to recognize liver tissue. If you ever wonder what those numbers are in the illustration, it is a key for us to identify what part is that. So let me tell you what those numbers stands. Number 1 stands as the central vein. Number 2 is the radiating cords of hepatocytes. Number 3 to 5 is what we call the portal triad. And under portal triad, we have the portal vein, hepatic artery, and the interlobular duct. So this is liver in a panoramic view. This time, let us learn about the liver in high magnification. In illustration A, it shows high magnification of liver as seen in drawing. In illustration B and C, it shows high magnification of liver in photomicrograph. On high magnification, the lobule is made up of polygonal liver cells arranged in the form of radiating cords. The central round nucleus of hepatocyte is surrounded by abundant cytoplasm. This description can be seen in illustration A. The cords are separated from each other by spaces called sinusoids. As I have discussed earlier, sinusoids are spaces between liver cells, and these spaces separates the cord from each other. The sinusoids are lined by endothelial cells and Kupffer cells or macrophage cells. In illustration C, it shows magnified view of portal tract. Each portal tract contains a hepatic arteriole portal venule, and one or two interlobular bile ducts. Normally, these structures are surrounded by fibroconnective tissue and a few lymphocytes. So, let me discuss or let me tell you what those numbers stands for. Number 1 stands for hepatic arteriole, number 2 for portal venule, and number 3 for interlobular bile duct. So the letter H stands for radiating cords of hepatocytes, and letter S stands for hepatic sinusoids. So this is liver on high magnification. Liver is made up predominantly of liver cells or hepatocytes. Each hepatocyte is a large cell with a round open-faced nucleus with prominent nucleoli. Liver cells are arranged in the form of anastomosing plates, one cell thick, and that the plates form a network in the spaces of which sinusoids lies. You can see it in our illustration. And for addition, when we say anastomosing, it is a cross connection between adjacent channels, tubes, fibers, or other parts of a network. In this way, each liver cell has a sinusoids on two sides. The sinusoids are lined by an endothelium in which there are numerous pores or fenestrae. A basement membrane is not seen. Interspersed amongst the endothelial cells, there are hepatic macrophages, or what we call the Kaffir cell. As you can see, the provided illustration shows the relationship of bile capillaries to liver cells, and this is just a schematic representation. Bile canaliculi, also known as bile capillaries, are thin tubes that receive bile secreted by the hepatocytes. The bile canaliculi eventually merge and form bile ductules. The bile passes through the canaliculi to the hepatic bile ducts and then into the common hepatic duct, which drains directly into the duodenum.
Now, the sinusoid space of these, also known as the perisinusoidal space, is a location in the liver between a hepatocyte and a sinusoid. It contains the blood plasma. Microvilli of hepatocytes extend into this space, allowing proteins and other plasma components from the sinusoids to be absorbed by the hepatocytes. As you can see from our presentation, from the upper left, the sinusoidal surface. The sinusoidal surface is the cell surfaces adjoining sinusoids, bears microvilli that project into the space of these. The cell surface here also shows many coated pits that are concerned with exocytosis. Both these features are to be associated with active transfer of materials from sinusoids to hepatocytes, and vice versa, and about 70% of the surface of hepatocytes is of this type. Next is the canalicular surface. Such areas of cell membrane bear longitudinal depression that are opposed to similar depressions on neighboring hepatocytes to form the wall of a bile canaliculus. Irregular microvilli project into the canaliculus. On either side of the canaliculus, the cell membranes of adjoining cells are united by junctional complexes. About 15% of the hepatocyte surface is canalicular. The intercellular surface. These are areas of cell surface where adjacent hepatocytes are united to each other just as in typical cells. Communicating junctions allow exchange between the cells. About 15% of the hepatocyte surface is intercellular. Now, as you can see, the yellow part in our presentation, or the bile canaliculae, also known as bile capillaries. These are thin tubes that receives bile secreted by hepatocytes. Now, the surface of the liver cell is separated from the endothelial lining of the sinusoid by a narrow perisinusoidal space. Microvilli present on the liver cells extend into this space. As a result of these factors, hepatocytes are brought into a very intimate relationship with the circulating blood. Some fat cells may also be seen in the space of these. Now, endothelial cells form a single cell layer that lines all blood vessels and regulates exchanges between the bloodstream and the surrounding tissues. Signal from endothelial cells organize the growth and development of connective tissue cells that form the surrounding layers of the blood vessel wall. The fenestration in endothelium facilitate an easy access of nutrients, drugs, xenobiotics, and toxins in the sinusoidal blood to hepatocytes. Tight junction form the continuous intercellular barrier between epithelial cells, which is required to separate tissue spaces and regulate selective movement of solutes across the epithelium. The hepatocytes have many microvilli which project into this space to increase absorption from the plasma. The space of these also contains lipocytes that store fat and vitamin A. They can also become contractile, and they make the type 3 collagen fibers, also found in the space of these. Again, microvilli of hepatocytes extend into this space, allowing proteins and other plasma components from the sinusoids to be absorbed by the hepatocytes. Now, at this part, we will tackle about the gallbladder. Gallbladder is a muscular sac situated on the visceral surface of liver in the fossa for gallbladder. The gallbladder stores and concentrates bile. This bile is discharged into the duodenum when required. The wall of the gallbladder is made up of a mucous membrane, a fibromuscular coat, a serous layer that covers part of the organ. Now, as you can see in our presentation, we've labeled it, and number 1 represents the mucous membrane lined by tall columnar cells with striated border. Number 2 is the lamina propria, and number 3 is fibromuscular coat, and 4 is the serosa, and the letter C is crypt in lamina propria. So as we continue, the muscular membrane, or mucous membrane rather, of the gallbladder is lined by a tall columnar epithelium with a striated border. The mucosa is highly folded. In sections, the folds may look like villi. The gallbladder mucosa is second only to the kidney in its absorptive capability because of its concentrating ability. The gallbladder removes a large amount of water and ions. This situation allows previously unsaturated bile to reach higher cholesterol saturation indexes. The fibromuscular coat is made up mainly of connective tissue containing the usual elements: smooth muscle fibers are present and run in various directions. And the serosa, the serous layer, has a lining of mesothelium resting in on connective tissue. The fundus and lower surface of body of gallbladder is covered by serosa, whereas the upper surface is attached to the fossa for gallbladder by means of connective tissue or adventitia. Now, it contains many elastin fibers, lymphatics, and in the neck of the gallbladder gland which secrete mucus. The lymphatics of this layer help to drain water when the bile is concentrated, and the mucus glands may create a surface that protects the wall of the biliary tree. At this slide, it shows the schematic representation section through the major duodenal papilla to show the components of the sphincter of OD. The hepatopancreatic duct, at its lower end, the bile duct is joined by the main pancreatic duct, the two usually forming a common hepatopancreatic duct or ampulla that opens into the duodenum at the summit of the major duodenal papilla. The mucosa of the hepatopancreatic duct is highly folded. These folds are believed to constitute a valvular mechanism that prevents duodenal contents from entering the bile and pancreatic ducts. So the ampulla is surrounded by the sphincter of OD, which not only controls the flow of bile and pancreatic juice into the duodenum, but also prevents the reflux of duodenal contents, bile and pancreatic juice into the bile and pancreatic ducts. The sphincter of OD, well-developed smooth muscle, is present in the region of the lower end of the bile duct. This muscle forms the sphincter of OD. From a functional point of view, this sphincter consists of three separate parts. The sphincter collidicus surrounds the lower end of the bile duct. It is always present, and its construction is responsible for filling of the gallbladder. A less developed sphincter pancreaticus surrounds the terminal part of the main pancreatic duct. A third sphincter surrounds the hepatopancreatic duct or ampulla and often forms a ring round the lower ends of both the bile and pancreatic ducts. This is the sphincter ampullae. The sphincter ampullae and the sphincter pancreaticus are often missing. The pancreatic duct, as you've seen in the slide, it is composed of pancreatic islets, which appear as islands of cells dispersed between the pancreatic acini. These islet cells produce and secrete hormones that regulate glucose, lipid, and protein metabolism. The sphincter regulates the secretion of pancreatic juice and bile into the duodenum. It also prevents reflux of duodenal contents into the ampulla of Prater. At this slide, at this part, as you can see, we've labeled the drawing, and the number 1 represents the serous acini inside lobules. Number 2 is the islet of Langerhans, and number 3 is the intralobular duct. Number 4 is the interlobular duct. The exocrine part of the pancreas has closely packed serous acini similar to those of the digestive glands. It secretes an enzyme, rich alkaline fluid, into the duodenum via the pancreatic duct. Pancreatic islets, also called islets of Langerhans, are groups of cells in your pancreas. The pancreas is an organ that makes hormones to help your body break down and use food. Islets contain several types of cells, including beta cells that make the hormone insulin. Intralobular ducts have a classical cuboidal epithelium and, as the name implies, are seen within lobules. They receive secretions from intercalated ducts. Interlobular ducts are found between lobules within the connective tissue septae. They vary considerably in size. The smaller forms have a cuboidal epithelium, while a columnar epithelium lines the larger ducts. Intralobular ducts transmit secretions from intralobular ducts to the major pancreatic duct. Now, as you have seen, this is a gland made up of serous acini. The cells forming the acini of the pancreas are highly basophilic, or like bluish staining. The lumen of the acinus is very small. Some acini may show pale staining in the center. Among the acini, some ducts are seen. The ducts have a distinct lumen lined by cuboidal epithelium. At some places, the acini are separated by areas where we see aggregation of cells quite different from those of the acini. These aggregations form the pancreatic islets, pale staining cells arranged as groups surrounded by blood vessels. In the photomicrograph, an interlobular duct lined by cuboidal epithelium is surrounded by lobules of acinar cells, which have small basal nuclei and granular eosinophilic or slightly basophilic cytoplasm. Islets of Langerhans appear as groups of small cells with lightly stained cytoplasm.